friends, so to start with the next lecture, that is DNase treatment. DNase, why you need DNase treatment in molecular laboratory? You want to get rid of your DNA contaminant from the RNA sample. You have isolated RNA sample either from kit based method or manual method or combined method. Your DNA is a major contaminant in the sample. So you have to remove that DNA from the RNA sample. So for that we can utilize DNase I enzyme. This enzyme, it will make cuts, random cuts. It will make random cuts, and it has 5-phosphate and 3-OH, 5-phosphate 3-OH groups at 5' and 3' ends. So like that, random your fragments will be generated, or your DNA is chopped into small, very small fragments which cannot be used for any PCR, or which cannot be utilized as a template for the PCR, and it will not interfere your gene expression studies. So DNase I treatment is important while we have to do gene expression studies, or secondly when we are going to for in vitro transcriptions, and when we have to like you have to RNA footprinting you have to perform, or you have to prepare cDNA libraries. In all those cases you have to get remove of your DNA. So where we need DNase treatment or pure RNA population? Where? In gene expression studies. In gene expression studies you need pure RNA. Second, in vitro transcription. In vitro transcription. Third one is in cDNA library preparations. cDNA library preparation. And fourth one is RNA footprinting. RNA footprinting. So in all these cases you want to have pure population of RNA. So whenever you are isolating RNA from any method, so always DNA is contaminating your RNA. Okay, so DNase treatment is important. Now the component of DNase treatment, there are two different type of buffers are provided in DNase along with DNase I enzyme. One is 10x buffer with magnesium ion, and the second one is 10x buffer with manganese ion. So both the ions, they have different preference. So how it will be utilized? Let us see. So if you are using magnesium ion and if you are using manganese ion, so how it will help? So if we see DNA, this is your DNA molecule. So in presence of manganese, it will make random cuts here, there, here. So you have some very small fragments with 5' phosphate and 3' OH groups. But in presence of manganese, this is your DNA. So in presence of manganese, it will make exact cut at the same site of both the strands. So it will generate your exact fragments, very small fragments, but of both the strands are of equal size. It is not random. Here the cut or the digestion is random. Here the digestion is not random. It is exactly blunt and small fragments. Now the question is where we are using magnesium ion and where we are using manganese ion. So magnesium ions are used where you don't want any DNA molecule to be as a contaminant in your RNA molecule, like gene expression studies, or if you want to go with the cDNA libraries, or if you want to go with the in vitro studies. But where we have to use manganese? If you want to prepare genomic libraries or DNA libraries, there you want some equal fragments then the DNA will be treated with the DNase I in presence of manganese ion, and it will generate small fragment which is utilized for the DNA library preparation. So these are the two different ions which have different type of properties to make the DNA digestion. Now, what is the procedure of DNase treatment? How you have to give? The company they provide DNase I as 1 unit per microliter. That means if you are using 1 microliter, it has 1 unit. And as you know that if you are using 1 unit of, if you are saying 1 unit of any enzyme is 1 micromole. How you can explain the activity of enzyme in terms of units? So 1 unit of enzyme is that amount of enzyme which convert per minute. What is 1 unit? 1 unit is the amount of enzyme which convert 1 micromole of your substrate, per minute. जो enzyme है, अगर मैंने बोला 1 microgram of your plasmid you are using, and you want to digest that plasmid with the DNase I, that means 1 microgram of plasmid should be digested per minute. That is your enzyme activity. So, but 1 unit means whenever you are using, if you are taking 1 microgram of your RNA, considering that it has good amount of DNA, 1 microgram of RNA, and you want to treat this 1 microgram of RNA with the DNase I, you have to use 1 unit of DNA. 1 unit of DNase I. Right? So, for the treatment of RNA, what you have to do? You have to take 1 microgram of RNA. ठीक है? 1 microgram of RNA आपने ले लिया, उसमें आपने 1 unit of DNase I डाला, प्लस उसमें आपने जो DNase I का बफर था, DNase I बफर डालना है in the concentration of 1x, 10x का आपको 1x करना है, and then you have to keep at 37 degree for 10 to 15 minutes. Why we are giving 10 to 15 minutes? Because we want to give sufficient time to degrade your DNA as a contaminant. तो आपको DNA को remove करना है. In that case, you have to give sufficient time so that DNase will degrade all DNA molecule present in your RNA, eluted RNA. So, what you have to think? You have to give 10 to 15 minutes at 37 degree. Now, you have given sufficient time to degrade your DNA from the RNA molecule, and how much amount of RNA you have used? 1 microgram of RNA. Now, you have to inactivate your DNase I. You have to inactivate that also. So, the inactivation is very simple. You have to use 1 microliter of 25 millimolar EDTA. EDTA is a ion chelator, so it will take out all the magnesium ion which is required for the DNase I activity, and hence the DNase activity is inhibited. So 1 microliter of 25 millimolar EDTA you have to add to that solution. Then you have to heat the tubes at 75 to 80 degree for 5 minutes. Just to further degrade your DNase I. EDTA ने उसकी एक्टिविटी को खत्म किया, बट उसको इनएक्टिवेट, कंप्लीटली इनएक्टिवेट कौन करेगा? DNase I को, आपका हीटिंग प्रोसेस. So both step you have to follow. You have to give 1 microliter, you have to provide ion chelator also, and then you have to heat the sample to inactivate, complete inactivation of your DNase I. So 1 microliter of आपने EDTA डाला, then आपने अपने DNase को inactivate. क्यों किया? Why we require the DNase inactivation one? Because in next step, whenever you have to convert your RNA to cDNA, the cDNA will be target of DNase I. It can degrade your DNA. So this is the treatment and inactivation whole process of DNase I treatment to remove your DNA as a contaminant from the RNA sample. So this is the process, and very simple. You have to include in your protocol if you are going with the any gene expression studies. So thank you, and if you have any questions, you can write to me. You can ask me. If you want any detail, you can write to me regarding the DNase I treatment, regarding any method, RNA isolation method, manual, or your kit based method, or your combined methods for tissues, for cells, for anything. If you want other body fluids such as nasopharyngeal swab samples, throat swabs, or your oral swabs. If you want to have any question, if you want to ask any question, you can write to me. Thank you.